Hi, everybody. Our today's lecture is going to be focusing on the process of gene expression and how we go from DNA to our proteins. And we also talk about how those proteins that have been synthesized by the ribosome undergo changes in their folding to create this beautiful 3D structure that ultimately determine the function of the protein. So since we are going to talk about proteins, the first thing I want you guys to understand is proteins, again, are macromolecules. And these macromolecules are one of the main categories of the four macromolecule or organic macromolecules we have. And as we talked about previously, we talked about nucleic acids, I shouldn't be writing that high. Sorry, nucleic acid. And we said if that's our macromolecules, the smaller units are called nucleotides. And then if we're talking about proteins in this case, if we apply the same concept, the little units or subunits that make it are amino acids. In reality, nucleotides, there were only 4 types when we're talking about the DNA. And in case of the proteins, we actually have 20 different type of amino acids. And again, the sequence of that amino acid basically going to determine the proteins that we are going to create. Now, when we're looking at these 20 amino acids, if we to categorize them, 9 out of those 20 amino acids are going to be referred to as essential. So what does that mean? Essential means that we as humans are unable to make these amino acids by ourselves, and therefore we need to get those amino acids directly from our diet. So if you have an unhealthy diet where you didn't get a balanced nutrition in you, you may have problems with protein development and condition that arises that has to do with protein synthesis. What foods are typically rich in amino acids or these essential amino acids? So the dairy products are pretty good: egg, fish, quinoa, beef, peanut butters, and almonds. So it goes basically range from if you are, you can eat anything, to vegetarians to vegans. It covers everything within that range. But regardless, we can get those sources either from other animals or the products of the animals or the products of the plants. Now, we're talking about the fact that there are 20 different amino acids. But if we look at those amino acids, are they actually a significant difference between them? And the answer is yes and no. So let's take a look at this. When we take a look at the amino acid, which is, again, an organic molecule, right? You're kind of hopefully recalling, organic molecule implied that it has to have carbon and hydrogen as their part of their structure. So here, this is our centralized carbon right here. And you notice that at one hand is connected to a single hydrogen. So that's fulfilling the requirement for carbon and hydrogen. You also have 2 additional groups that are going to be consistent regardless of which of the 20 amino acids we're looking at. So this is the one you see as the amino group, which is, if I was to write it, would be NH2. And then you also have on the other side, you have a group called carboxyl group, which is COOH, which is again the carboxyl group. And if you are again comparing the 20 amino acid, this portion, so this section, and this section remain the same. So the only piece that is basically differentiating or distinguishing these 20 amino acids from one another would be what we consider the R side chain. So the R group or the R side chain is what distinguishes those 20 amino acids from one another. Here is the picture of what those 20 amino acids looks like in terms of their R group. Notice that they have different characteristics by the color coding we show here. So we have some that have the either positive or negative charge. So this is our negative charge, this is our positive charge. There are ones that have uncharged but polar side chains or R chains. And there are some special cases. And then the most of the 20 amino acids are identified as amino acid that has the hydrophobic characteristics. And again, hydrophobic implies that they do not like to interact with water or they're water fearing. So this is important in terms of how does your protein complete this final 3D structure, because it's relying on a lot of these characteristics to either form interactions or stay away. And again, those interactions or lack of interactions would lead to a very specific 3D structures. Now, in reality, if you look at the human body, we have about 20,000 different types of proteins within our body. And again, when you think about proteins, it includes roles such as the following. They can act as enzymes, which is basically what catalyzes the reaction. So when you use the word speed up, we can also replace it with the process of catalyze. You have support role, which is things like your hair, nail, skin that contains significant amount of a protein called keratin. We also have ability to transport other molecules with it. For instance, hemoglobin is responsible for carrying our oxygen molecule, and LDL and HDL is carrying our cholesterol, sorry. And again, I mentioned this before, but LDL is usually what we refer commonly as bad cholesterol, and HDL as the quote unquote good cholesterol. Another thing that you may have heard of it, especially after what we have gone through the past few years, is antibodies. Antibodies are basically these proteins produced by your white blood cells, and they're responsible for fighting off any pathogens, which include parasites, bacteria, viruses, etc. A lot of our hormones are also identified as proteins. The two main ones that we continuously hear, if you have ever taken biology class, is insulin and then the growth hormone. Insulin is extremely important for maintaining your blood glucose level. And if it's not maintained, it can lead to diabetes. And growth hormone is very active in children and also for us to maintain our health, basically. Regulation and signaling is another main thing. This is not something we per se see on our day-to-day activities, but it happens in a cell every single second. And this is basically what we use a messenger system where one protein signals another one, and signals another one, signal another one, that there is this cascade pathway that allows us to either regulate a specific activity in the cell, either upregulate it or downregulate it, or sending signals for a specific action. Motion and mobility, I think, is very easy to remember, because when you think about muscles, the first thing that should pop into your head is your skeletal muscles that gives you the ability to move. But it's not limited to that. We also can include our cardiac muscle, which is basically helping your heartbeat. And then we also have your smooth muscle, things that help the movement of your stomach or your intestine. So the digestion of your food highly depends on the type of movement. Metabolism is another one. Again, it is important in terms of you taking that glucose molecule and producing your ATP, or taking proteins or fat and converting it to ATP. Protection and repair. Again, when we're looking at if you cut yourself or there's some sort of bleeding, we have what we call clotting factors as well as repairing factors, such as fibrinogen, that come into the play, clotting factors, and basically these are proteins that work together to make sure that you do not lose significant amount of blood, and then also repair the tissue as time passes by. Ion channels. Ion channels are very important. These are things like potassium channel, chloride channel, calcium channel, etc. They are very specific to each of the ions. And again, it controls what gets in or out of your cell. Fluid balance is another one. It's important in terms of making sure the amount of water that stays in your blood is consistent. If you have problem with these proteins, specifically called albumin, if you have too little of them, then the water moves into your tissue and causes what we call edema or swelling. And then if you don't have enough if you have too much of it, the water gets pulled out of your tissue and into your blood and may cause hypertension. Digestion, you have a lot of enzymes that are basically proteins involved in the process of breaking down your large macromolecule groups to single units. And lastly, you also have pH regulation that's associated with your proteins that ensure basically your blood pH maintained at a very specific number between the range of 7.35 to 7.45. So it took me a long time to talk about this, but it also kind of highlight the importance of having proteins and the role that proteins play in our body. If you look at the carbs and lipids, as we mentioned before, they are going to be the main source of energy. And proteins are going to be the one that use up that energy to basically carry out the regular functions of the cell, tissues, organs, organ systems, and eventually the whole body. So proteins, basically very important for us. Now, again, recalling from our first lecture, I believe, when we're talking about the protein, each of them have its corresponding gene. So basically you have a gene on your whole sequence of DNA, and that gene is encoding for a very specific protein. And if you're looking at all of the DNA that encodes for genes, we refer to that as the genome, the individual genomes. And to, again, recall that information, the genome, you receive a genome set from your mom and it's set from your dad. And the more specific thing that I wanted to talk about, and this is something that's going to come up as we go through this lecture, is there are only parts of the gene that basically encodes for proteins, and those sections are called exons. And we'll discuss that again in terms of introns and extrons as we go through the lecture. Now, when we're talking about proteins, we already understood that proteins are made up of amino acids. And when you put hundreds or thousands of these amino acids together, we create what we call proteins, or a long sequence or chain called a polypeptide. Polypeptide simply means number of peptides, which is again referring to your protein. Now, each protein has a very specific sequence. So if you're looking, for instance, going back to what I was talking about, the protein sequence for your insulin versus the protein sequence for the growth hormone, just looking at the sequence, they are very different. So the sequencing of the amino acid would be different. There are also going to be different number of amino acids that make up the protein. So one protein may use only 1,000 amino acids, the other may use 1,500 amino acids. Some might be super small and they use 200 to 500 amino acids. So sequence of the amino acid plus the number of amino acids that are utilized to produce your proteins basically are determining factor of how those proteins are going to become what they are. Now, a chain of polypeptide proteins, again, many amino acids line up together, are eventually going to fold based on their sections, based on the type of bonds that they can create, and eventually create a very beautiful 3D structure, and that's referred to as its conformation. So conformation, or the shape of that protein, basically determines its function. So if it's shaped like A, that means it's an insulin corresponding to maintaining your blood glucose. And if it's shaped like protein B, that implies this is a growth hormone and it's essential for the ability to affect different organs and therefore help you with the growth of the body. Here is basically what I'm showing you and hoping you guys understand. On both of these images, we're showing the same thing. I just used a kind of a different coloring and 3D images. So on the top, you have the first line, which is representing the sequence of the amino acids. This is what we call primary structures. And by the way, I will go into more detail about this. And then as the complexity of the structure starts to form by forming those 3D structures and fold, you form helices and beta sheets, which is demonstrated here. And then you put everything together and you create what we call a tertiary structure. And if the tertiary structure needs additional component added to it, then you create what we call a quaternary structure. Now, again, quaternary structure demonstrated on either here or here. Both are depicting a very complex 3D structure, and that structure or conformation determines what that protein will be doing. Now, we already have discussed again this on our previous lectures when we described how does your gene goes from being a gene and a DNA to becoming a protein with a very beautiful 3D structure and a very specific function. And that process of the gene expression happens at two levels. The first level is a process of transcription, where you take the original DNA sequence and you make copies of that gene in a form of mRNA, which is also a nucleic acid. And then the second part of that process is taking that RNA sequence and taking it to the ribosome within the cytoplasm of the cell, and using that RNA as a print, as a blueprint to make or manufacture our proteins. And then finally, that ends with, again, that folding of going from primary to secondary to tertiary and possibly quaternary structures to form the final conformation of the protein. Now, when we're looking at this concept of transcription and translation, the concept of central dogma comes into play, which basically means genetic informations always follow one direction, which means you first take the DNA, and DNA forms RNA, and then RNA is utilized to make protein. If you have organisms that only have the RNA and no sequence of DNA, and there are organisms like that, then you also go from RNA to protein. But regardless of which one of these two we use, we are always going into one direction. So proteins, for instance, are never going to be used to make RNAs. RNAs will never be used to make DNAs. And that's basically what central dogma implies. Now, nucleic acid. Again, we have talked about nucleic acid in details. We have talked about DNA more specifically in details. Just to kind of highlight what we already discussed, DNA is a macromolecule made up of nucleotides. These nucleotides have a sugar phosphate backbone, and they also have an option of one of the four bases. That is either the A, T, C, or G, which stands for adenine, thymine, cytosine, and guanine. Now, DNA, as we have learned, is important in terms of storing the genetic information that eventually will be used to encode for proteins. But the issue is that DNA, there's only one copy per cell, so it has to be protected and make sure when it's utilized, it's in the safest form. RNA, on the other hand, is a similar structure to your DNA. However, one of the main differences, and I will talk about more of these differences, is that the sugar phosphate backbone that the RNA has is made up of a sugar called ribose versus a sugar for DNA would be deoxyribose. But again, RNA is important for us to carry out the process of translation, where these copies of the original gene was produced and moved out of the nucleus into the cytoplasm for the purpose of producing and utilizing it for protein synthesis. Now, here's are some of the main differences you will see. There is, of course, always exception to the rule, but this is a general terminology and concepts that we utilize to highlight the difference between the DNA and RNA. First, DNA is usually double-stranded, as you can see from the bottom picture on the right-hand side, on the left-hand side, and then RNA would be only single-stranded. And again, this is usually. The DNA bases, you have, again, A, T, C, G, but in terms of RNA, the T will be replaced with the letter U, which stands for uracil base. So that's another main way of us distinguishing if we're looking at a DNA sequence or an RNA sequence. Deoxyribose, as I mentioned before, deoxy implies lack of oxygen. So deoxy means lack of oxygen in that sugar. And in terms of RNA, the sugar does have that oxygen and therefore referred to as ribose. DNA has the ability to maintain protein encoding information, while RNA basically takes those protein encoding information and controls how that information will be used by the ribosome for the synthesis. And DNA, again, we only have one copy per cell, so it persists inside the cell. We don't destroy it, we keep it. Versus when we talk about the RNA used for the process of translation, they are short-lived, and as soon as you're done making the copies, making the proteins, then the RNA will be degraded. Here is another visualization in terms of the double-stranded versus single-stranded. You also can see, you can also see the presence of the oxygen versus the lack of it in the DNA. And also the differences between the thymine versus the uracil, which if you're looking at it, they are very close to each other. It's the lack of 2 hydrogen that makes the difference between the thymine versus the uracil base. So let's take a look at the process of transcription. But first, for us to understand the process of transcription, we need to understand some of the vocabulary. So as we go through the process of transcription, which again, taking the DNA sequence and making an RNA sequence from that, we need to open up the double helix, and the double helix, the strand that is going to be used for the mRNA to be making its, for making of the mRNA, we refer to that as the template strand. Now, for the process to happen, very similar to the DNA replication, where we use a polymerase to add the bases, we can use the same concept here. The difference is for replication, we use the DNA polymerase. In the process of transcription, we're going to be using the RNA polymerase. Now, non-template strand of the DNA within that double helix is called the coding strand, which basically going to have the same sequence as your RNA with a replacement of U instead of a T. So here is what that process looks like. Again, here is the original template DNA sequence that are being utilized. So that is the gene that we want to make basically. And the green sequence that's being produced, that's your mRNA. Sorry, RNA. I haven't talked about the M portion of it. And again, notice that this process is also, again, 5' to 3', just like the transcription process and replication if you do compare those two together. The second strand, which is basically, again, going to be identical to the RNA sequence being synthesized with the difference of a T and U replacement. So again, when I talked about the RNA, I have mentioned it a couple of times, as I mentioned this copy of the DNA as mRNA. So mRNA stands for messenger RNA, which is, again, the one that's going to be utilized to make our proteins. Now, we also have other type of RNA. The first one that is extremely important for us is called ribosomal RNA, or rRNA. This is the one that basically going to form the structures of the ribosome organelle to be utilized during the process of protein synthesis. And we also have tRNA, or transfer RNA, which is a really small structure, but extremely important, because at one end, it will be connecting to the amino acids that are being utilized to make the protein sequence. And on the other end, it would be connected to the RNA and matching what we call an anticodon with the original codon of the mRNA. If the codon, anticodon does not make sense, or you have never heard of it, that's okay. We will expand on this as we go through the process of translation. So here is what we're discussing in terms of mRNA. mRNA is important in carrying the genetic information to make very specific proteins. They are, the mRNA that's being produced is going to be read or processed during the process of translation, 3 at a time, and those 3 are at the time are referred to as the codon. So for instance, on this one, if you're looking at your mRNA, these three are one codon, here's another codon, here's another codon, and here's another codon. Now, why is this codon so important for us? Is because these codons basically tell us which amino acids are going to be synthesized from that codon, or which amino acid is going to be en route to be placed to make my protein. Now, mRNAs can be relatively short, which is about 500, and it could be as long as 4,500, which makes you about a 1,500 amino acid protein. So you can divide it by 3 because they read in sequences of 3 or 1 codon. Now, do notice that there are cells that are differentiated cells, which is basically cells that have undergone the process of differentiation. They have certain groups of mRNA that are going to be transcribed. They're not going to do everything. So not all of the genes basically would be active in every cell in your body. And again, that transcription will eventually encode what proteins are going to be synthesized. And those proteins would be the ones that are super important or important for the functionality of the cell in a very specific location of your body. For instance, the cells that are being transcribed and translated in the heart will be different from the ones transcribed and translated in your skin versus the ones translated and transcribed and translated in your brain. Now, again, we mentioned that concept of ribosome and the organelle ribosome. That is something that described in your cell lecture for the second week. And the RNA components, basically, the ribosomal components are coming from an area of the cell called the nucleosome, which is inside the nucleus. Most of the RNA can be relatively small, so about 100 nucleotides up to 3,000 nucleotides, so they could be large. And as you can see, there are typically what we talk about RNA utilized for ribosome. It creates 2 subunits, one that's considered the small subunits, and the second one is what we consider a large subunit. So by using these 2 subunits, putting them together, we are able to take the mRNA sequence and connect and create our proteins. Now, RNA is very important for structural support as well, and they are going to sometimes they can act as catalysts and help the process of aligning the ribosome with mRNA. So this is not the only rRNA that we have, which forms the ribosomes. There are additional components that allows you to do that too, in support of it. Now, I find tRNA to be very, very interesting RNA. First of all, again, T stands for transfer RNA. If you notice this is the orange, what we have here is our tRNA. At one side, it says amino acid attachment site. And the other side, it has the mRNA. So on one side, it's going to basically connect to the mRNA. And this is what I want you guys to notice. On the mRNA, you have your codon. So here's your codon, here's the second codon, here's another one, and another one. On the second side of the tRNA, we have what we call anticodons. Anticodons literally means they are the ones that are going to be pairing with our codon. So in this case, G pairs up with C, A pairs up with U, and G pairs up with C. Now, here's the thing. This anticodon, but you have this anticodon in this side, it gets a very specific amino acid on the other end. So if you have CUC, you get, let's say, amino acid number 5. If you have GUG, then you get amino acid number 7. So the anticodon that matches the codon, again, I'm going to say that, the anticodon of the tRNA that matches the codon of the mRNA has a very specific amino acid attached on the other end of it, because those sequence of amino acid is what allows us to synthesize a protein. Now, this this RNA is relatively short. It's only about 75 to 80 nucleotides. So compared to the other parts where you have 500 to 1,000 to 1,500, this is relatively small. Here is a depiction of 3 separate tRNA and what I meant by having a different amino acid attached to each of them. So on the light green top line, you have the mRNA set in the codon. So again, you have 3 here, 3 here, 3 here. The dark green side, which is these ones right here, those would be your anticodon. And the anticodons are connected to my tRNA, which is shaped like a T, right? Now notice, as the sequence of the anticodon are different here, the amino acids that are attached here are each different. Now, does it mean that every anticodon will have a different amino acid? And the answer is no. But through years and years of research, we have figured out what codon gives us what a specific amino acid on the other end. So if we have the UUU or UUC, it's going to encode for phenylalanine. If you have UUA and UUG, it gives us leucine, and so on and so forth. So to help you understand this, these are your 3 codons, 1 on each side, right? So UU and then U, so it gives you UUU. And then U, and then C on this side gives you UUC. So the letters are here. That gives you your codon, right? And what you have in front right here would be the amino acids for those codons. I want you guys to know 2 things here. So there is 3 stop codons. So these 3 are very important. So if you have UAA, UAG, and UGA as an amino acid, that automatically terminates a sequence of amino acid, which means that ends the protein. So if you have any type of mutation that leads to placement of the stop tRNA anytime within the amino acid, it would literally break that amino acid and it leaves it incomplete. And then the other one is the MET, which is a start for the methionine, which is a start codon. And we always have, regardless of what protein I'm making, it's always going to start with AUG, because there is only and only one start codon that initiates any amino acid being connected and initiate any protein synthesis within our body. It's always going to be AUG. Now, we're talking about, again, the process of transcription and then using the mRNA created to make our new protein. So what are the components needed for the transcription? So just like replication, where we have factors that came in and were involved in the process, we also have transcription factors. So transcription factors are basically responsible for interaction and forming the beautiful apparatus you can see at the bottom, which are these little 4 circles, allow the DNA to, they bind to a specific part of the DNA that is referred to as a promoter, and allows basically or targets or initiates the process of transcription. So without this transcription factor, or if you have any issues with this transcription factor, you basically can't even think about the process of transcription. So it says it initiates transcription at a very specific site, which is basically initiating the process of transcription. Now, it does respond to signals from outside the cell. So it depends basically what your body needs or what cells within your body needs, what type of protein synthesized. And based on those signaling coming from messenger signaling, you decide what protein needs to be transcribed and therefore use the transcription factor to target the DNA and therefore initiate the process of transcription. Now, here's the thing, as I mentioned earlier, mutation basically changing anything in this transcription factor. So modification of this transcription factor may cause a wide range of effects, as in simple that I wouldn't see as much effect or changes. You would be relatively healthy versus major health conditions that can lead to death within a very short period of time. Now, for the process of transcription to happen, you undergo 3 main steps. The initiation, which is associated with your factors of transcription factors, the process of elongation, where you basically create your mRNA sequence using your DNA sequence, and then termination, where you basically finish up the process of transcription, make sure everything is nice and neat, and get it ready for it to be sent out of the nucleus for the process of translation. So here is the initiation process, something we already seen. Initiation is where your transcription factors connect. The transcription factor help RNA polymerase, the one that's going to allow the elongation of the mRNA, to come and bind to a very specific region. As I mentioned, that specific region is referred to as the promoter. So you cannot accomplish anything unless you start with your transcription factor, which opens the door for the RNA polymerase and initiates the process of transcription. Now, elongation, again, is pretty simple. The RNA polymerase reads the strand of DNA, and based on the DNA sequence, it adds a specific nucleotide. So these 3 nucleotides that are being used to synthesize your RNA are either U, A, C, or G, and are going to be added depending on the sequence of DNA. And then finally, when you finish with your gene, you've done whatever you need to do for transcription, you will terminate it by looking at, again, a terminator sequence, which usually happens, again, at the end with very specific pattern. And that when your RNA polymerase reaches that end, it stops adding polymerase. It is separated from your DNA sequence and therefore effectively terminate the process of transcription. Here is giving you an idea of what that process again looks like as we go through it. Beginning of translation, we have the promoter sequence where the cofactors are going to be binding to. One of the things that you do see as promoter is what the region we call a TATA box, which is a repetition of TATA. The binding factor that's depicted here is a transcription cofactor. And then here's another transcription cofactor that comes in. And as they attach themselves, RNA polymerase is eventually going to also come and bind to the region. And that initiates the transcription process. Notice as the double helix is opening up, the RNA polymerase is adding the individual amino acids, sorry, not amino acid, nucleotides to form your RNA. The RNA gets longer and longer, right? And eventually, when it reaches the end sequence, the RNA would separate and allow for that mRNA to eventually undergo changes and utilized for translation process. As I mentioned before, I do want you guys to remember that the mRNA sequence is always also created from the 5' to 3', which means that you are basically creating copy very similar to what you have done for your replication process. Another cool factor that I wanted you guys to understand is that your cell doesn't only create one copy of the mRNA at a given time. We need a lot of these mRNA because you want to have the ability to produce many proteins, many of the same proteins at a given time. One protein can never be enough for the cell, or one copy of that protein can never be enough for the cell. So we have what we call simultaneous transcription of the mRNA. And here you can see, again, this is the first sequence where the RNA polymerase initiated the process. Here's another one, and here's another one. So you have multiple amino acids basically synthesizing. So as the first one basically moves this way, right? Then the second one comes in, and move to the same direction, and then the third one moves in the same direction. So if you're looking at this is the one that first attached itself, this is the one that second attached itself, and this is the one that attached at the end, or the latest RNA polymerase that attached. So you're making 3 copies of mRNA based on this data, with number 1 being the one that has been going for the longest, so it's almost done with his mRNA. And number 3 at the other end is the one that just basically started the transcription process and still need to finish the mRNA synthesis. Now, the RNA that has been created so far, it's not done. It's not the final sequence that I want to use to make my protein or synthesize my proteins. So we do need to undergo some changes before that RNA leave the nucleus, go to the cytoplasm, and be used by the ribosome for protein synthesis. So what are those steps? Some of these steps include what we call a methylated cap, which is added at the 5' end. And this is what we identify as the recognition site for your protein synthesis. So this is always going to be the beginning of your translation process. What we call a poly A tail, which is added to the opposite end of the mRNA, or the 3' end of the mRNA. This is basically allow for the protein synthesis to begin and stabilize the mRNA. So you want to make sure basically it doesn't go anywhere while you're translating it. And then the last thing that I want to mention is something we already discussed in our first lecture about introns and exons. So remember that the whole sequence of the mRNA that's being created is not all going to be translated. We have regions that are considered to be intervening sequences, and they need to be removed. These are referred to as the introns. And then you have exons that are part of the mRNA that's going to be remain, eventually connected together and form the final sequence of the mRNA that will leave the nucleus and be utilized for the protein synthesis. Here is the picture that defines that concept and that explains the concept. So the blue sequence is the original sequence of the DNA. We have created an mRNA, which we refer to as the pre-mRNA. And notice that there are 2 introns and 3 exons. So this is not done basically. So first of all, we're gonna add our methylated cap at the 5'. We're gonna add the poly A tail at the 3', and then finally remove our introns from the sequence. So now you are going to end up with 3 exons, still having the cap and the poly A tails. And when this is done, this is what we call a mature RNA or mature mRNA. And you see it exiting the nucleus and getting into the cytoplasm. Now, a couple other things that I do want you guys to understand when we talk about RNA processing is that the introns and the exons of the RNA can be removed. It's a different section depending on the gene. There are different combinations of these exons being utilized to make different type of proteins. So if you say different combination of the exons of about 95% of the human gene encode different versions of the same protein, of a protein termed isoforms. So they're not necessarily, let's say, a very different protein. They're just slightly modified. So this mechanism of basically combining our exons at different sequences or different orders is called alternative splicing. So going back here at the last part of the picture, this process is the splicing. So if I change how it is spliced here, is basically what I determine what is the isoform going to be. Sorry guys, I dropped my pen. So let's take a look at the picture and see what that means. So the first picture I have here to identify protein A, you have 4 of your exon, a blue, a green, sorry, I should say 5 exon. Exon 3, 4, 5, right? So the first one has all 5 exons in order, which translated to the protein you see as protein A. The second one is an alternative splicing. You do not have all of the exon present. So you have eliminated number 3 on the second one. And this alternative splicing has given you protein 2. And lastly, you have protein 3, where in this case, number 4 exon, which is a relatively small one, is missing. And you get your last isoform as protein C. Again, notice that this is alternative splicing is giving you almost the same protein, but these proteins are slightly different. And the reason for this alternative splicing, before I forget, is that cell types actually use the same protein, but they may need a slightly modified version of it. And that's where the isoforms come into play. So you may, and don't quote me on this one, but let's say the protein A is used in the cardiac muscle versus protein B is used in a skeletal muscle versus protein C is used in smooth muscle. They're using in the cells, but the cells are different. So the protein is slightly modified to match the need of the cell itself. So we have talked about transcription. We just finished talking about transcription. We talked about DNA becoming an mRNA, mRNA undergoing changes, and those changes basically create a final sequence of exons that connects together and form what we call a mature RNA or mature mRNA. Now, this mRNA has now entered from the nucleus into our cytoplasm and ready to be connected to a ribosome and be utilized as a blueprint to synthesize its protein. So here again, the color coding shows that we have our original DNA sequence. We have our messenger RNA that has been divided into 3 nucleotide codons. And as we talked about, each of those codons encode for a specific amino acids. Now notice that they're showing amino acids are being linked together. So there's like amino acids on this side and amino acids on this side. And the linkage is a covalent bond that basically holds a very strong connection between, or creates a very strong connection between these amino acids. So let's take a look at some of the background information. In 1960, some of the researchers used a very logic, kind of common sense almost, and a clever experiment to decide and understand our genetic code. And based on those experiments, they found out that the genetic coding happened in triplets. So here comes the discovery of codon. Now, based on their information, based on the codons, they understood that there are 64 codons, okay? 64 codons. But those codons are only encoding for 20 amino acids. So don't forget that. Even though I have 64 codons, the codons only encode for 20 amino acids, which means multiple codons are encoding for the same amino acids. So again, I'm making these up. So let's say AAA and AAU both encode for the same amino acid, to just kind of give you an idea. Now, altering any DNA sequence that went 1 or 2 nucleotides, so you remove 1 or 2 nucleotides, cause major problem with the protein synthesis. This is referred to as reading frame shift. So you literally shifting the frame by 1 single nucleotide. So instead of you, and I think I have a picture here, if you shift it by 1, let's look at the original sequence. So you had GAC, 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 GAC. Everything looks good. If I have changed, or in this case, I think I've deleted 1. Sorry, I need to, okay. No, you added 1, sorry. I thought I posted a different picture. I added a G here. So now I have what? Instead of having GAC as my codon, now I'm gonna read GGA. The second one would be, instead of saying GAC, now it's reading CGA. Now, here's the thing. The codon looks significantly different, which possibly implies the amino acid they're connecting or the encoding will be very different. And you can see the GAC encodes for aspartame, and in this case, it's coding for glycine. Again, you were supposed to have aspartame. Now it's arginine. So you're significantly changing the sequence of amino acid by adding a single nucleotide base. What if I add 2? Now, again, instead of having GAC as my second codon, now I have what? UGG, which means I have added a U and a G and shifting everything by 2. So now, again, look at the original sequence. Everything was encoding for aspartame. Now it's completely different, right? Everything's changing. So remember that if the amino acid is different, then that causes the 3D shape to be different if anything forms. And if that's different, that means the structure of the protein is different. Hence, the function of that protein would be different. Now, notice the last sequence. If you add 3 bases at a time, what happened? You just added a leucine, but everything else stayed the same as aspartame, which means if I add 3 bases, I will not significantly change the sequence of the amino acid, which implies that the way your mRNA is being read at 3 at a time. So any shift less than 3 or more than 3 will cause a significant difference in the sequence of the amino acid being synthesized. Now, let's look at some of the characteristics of the genetic code. So it says, well, I guess this is non-overlapping, which means the same letter is not used for 2 different codons, something I mentioned before, right? You have 64 codons, but each of those specific codons only encode for a specific amino acid. It includes controls. So we do have a way for us to include direction for starting and stopping the translation, which we already talked about this. Those are the modification on your mRNA. And it is universal, which basically means life forms that are involved on the Earth are using the same structure, which means they have the same common ancestors. And different codons that are specified for the same amino acids are termed synonymous codons, and the ones that encode for different amino acids are called non-synonymous codon. And by that, if you want to kind of see what I mean, if I go back to what I was giving you the example here, actually, let me write it again so you guys have it on the same page. So synchronous codon, it says different codons that specify the same amino acid. So let's say again, arbitrary number, AAA and AAU both encode for lysine, right? So this is synonymous codon. But if I do this one, I said UAG, and it's encode for methionine, right? So this is a non-synonym coding sequence, which basically means it encoding for a different. The 2 on the top, synonymous codons, and then the one at the bottom, methionine, versus the other one on the top, they're considered non-synonymous codons. Now, we have described transcription, and we said, okay, we need the cofactors, we need the RNA polymerase, we need our nucleotides. So now we're going to go through the process of translation. What do I need to carry out the process of translation? So you need the mRNA, which kind of makes sense, right? You did all of these things in transcription, so you can make a copy and take that copy to synthesize your protein. We need our tRNA because tRNAs are going to supply our amino acids that are going to be added to form our proteins. And they come with a very specific codon, right? And then ribosomes, which are basically the organelle responsible for assembling the structures. And then for the amino acid sequence to be created, you need to create covalent bonds that hold them together. And for that process, we require energy on the form of ATP or GTP. I didn't mention GTP before. You don't need to know what it is. ATP is a main source. And then we also need what we call protein factors. Now look at the terminology, guys. Just like transcription, it has 3 phases: initiation, elongation, and termination. So the terminology is very similar. The difference is in transcription, we went from DNA to mRNA. In this case, we're going from mRNA to proteins. So translation initiation is basically relying on the leader sequence that is found in the mRNA that form a hydrogen bond with the small subunit. So here is basically what we call the leader sequence in dark green portion. This is a marker for the small ribosome to attach right after it. So a small ribosomal subunit, again, there's 2 parts. There is a small and large. So a small comes in and basically connects itself to the first part of the sequence on the mRNA. So you can see it, it's sitting on first codon, second codon, and third codon. So it's sitting on 3 codons right now. Now, to start the process of that, the next thing that we're going to add is our initiator tRNA. And this is the one that basically encodes for methionine, which is the start codon, regardless of what protein we're synthesizing. So you can see it coming right here and attaching to the sequence of AUG on the mRNA. And that forms your first and initial connection of the amino acid to basically start the process of translation. Now, this complex, which is a combination of the mRNA, the combination of the tRNA, and the small ribosomal subunit, is referred to as initiation complex. So there's 3 parts again: small ribosomal subunit, the tRNA, and the mRNA. Now, the next thing that you want to do is to add the large ribosomal subunits. So large ribosomal subunit comes in and initiates this really beautiful complete complex that we have, we're showing on a picture. So tRNA, as it comes in, the second tRNA comes in. So in this case, the tRNA is matching the codon GGA. So it's matching this codon GGA, and GGA encodes for glycine. So glycine comes in, and it forms a hydrogen bond. So here's the thing. It forms a hydrogen bond between the anticodon, which is these ones. These are my anticodons, and the codons of the mRNA. So right here, the connection between these two, the blue line that I'm drawing, that is made up of the hydrogen bond. So hydrogen bonds are not super strong, but it allows us to connect these 2 structures and hold them together. Now, notice that on the RNA, we have, sorry, on the ribosome, we have what we call an A site, and then we have what we call a P site. So at this time, the initial or the initiator codon, which is AUG, encoding always for methionine, sits on the first position, which is the P position or P site, and the second codon is sitting on the A site, which has connected to the tRNA with this corresponding amino acid. So 2 of the sites are now occupied. Now, as time passes by, this is really cool, you form a peptide bond. Again, this is required energy, which is a covalent bond. Peptide bond is a form of covalent bond. Okay, between the first amino acid, which is always methionine, and the second amino acid in this sequence being glycine. And when that happens, the tRNA that's now empty, right? It has dumped its methionine, it moves away, and glycine moves in to the position of P site. That means the A site is now empty. So what happens? The A site is now empty. So we look at the sequence of the codon of the mRNA. You have UGU for the mRNA, which means it matches the anticodon of the tRNA, which is ACA, and that's connected to an amino acid called cytosine. So cytosine comes in and occupies the A place now. Now kind of think about it. As cytosine comes in, what do you think is going to happen? Cytosine is going to form a bond with glycine, right? Which means this tRNA is going to fall off, and cytosine is going to move into the first space. Now the second space is now empty. We keep moving on this way. So now the next codon would be AAG. Find the corresponding anticodon and the tRNA connected to its amino acid. And that's exactly what happens. So you have methionine, glycine, cytosine moving to that. Lysine comes in next. Arginine comes in next. And again, every time they come in, they are going to connect with each other using a covalent bond. So covalent bond known as peptide bond. So by the time this process ends, you have what we call a polypeptide chain. Now, polypeptide chain ends when you reach our stop codon. Now, I don't know if you guys recall, but there are 3 codons that sequence or encodes for a stop codon. So here in this picture, we can see kind of the elongation happening, right? You only have like putting everything together. You only have methionine and glycine. Here's methionine, glycine plus cytosine. And then again, the continues. Here at this point, now we have reached what we call a stop codon. Here's our stop codon, which is a UAA. Now, instead of UAA actually connects to a tRNA and its corresponding antibody, not antibody, its corresponding amino acid, you actually connect to what we call release factor. And release factor literally say, oh, nope, I don't have anything attached to it. Nothing's gonna go this way, so that's ended. And that triggers the release of the full sequence of the polypeptide that has been created. As soon as the amino acid sequence falls off, the RNA and the large and small subunits also fall away. The mRNA goes its own way, release factor goes in its own way, and then the sequence of amino acid is now fully formed and ready to be modified. Here again is the depiction of the process, just like the transcription, where we don't do one transcription at a time, we do it simultaneously. We do the translation at the same way. So the one that you see at this end, this is the first one that attached, this is the first ribosome that attached, and the one you see at this end is the latest ribosome that has attached. So it barely has made its amino acid chain. Now, we talked about this before. I showed you a picture, but to just kind of discuss this in a little more detail. The protein structure that we have is a 3D structure, which means it requires folding. What we have as the polypeptide chain is just called the primary structure, which is literally all it is a sequence of the amino acid. If I start to form hydrogen bonds with the neighboring amino acids, I can create what we call alpha helices or alpha helix and beta sheets. And they look like this. So this is the result of the hydrogen bond. Again, you can either form these helices or what we call beta sheets, which looks like this. Kind of like when you make those paper fans. Now, if I take it a step further and I start to loop and coil these sheets and bend them using, again, hydrogen bond with the other neighboring carboxylamine groups, then I form even a more complex structure, which is what I see here. So that forms our tertiary structure. Now, at this point, this is already a 3-dimensional structure. But sometimes the protein that we make is really large, and it needs multiple tertiary structures to come in together to form a much larger protein. So in this case, if you imagine this is one subunit, so this is the subunit, this is subunit, right? So this is subunit 1, this is 2, this is 3, this is 4. So in this protein, which is a really large protein, you're actually combining 4 tertiary structures to make this much larger quaternary structure and holding them together to make, again, a much more complex, large proteins. Protein folding, it begins as the process of amino acid chain starts to finish. So as we're kind of coming to the end, the protein starts to fold. If the proteins are misfold, they are basically marked with a structure, with a protein called ubiquitin. And this protein, when there is more than one tag, they basically, you're gone. It's not good. they send it to a structure called proteasome. And proteasome is just like a tunnel. So let me show you this. This is the proteasome in purple structure. So you have a misfolded protein. It gets marked by ubiquitin. And if it is marked with ubiquitin, all you do is it goes through this proteasome, and this tunnel basically it is straightened and dismantled. So you unfold it and then you cut it to pieces. Proteasomes also destroy properly folded protein that might be basically too much of it, or your body doesn't need it anymore. So lots of antibodies undergo this process. So again, look at it. The first picture is you have a misfolded protein. It gets tagged by ubiquitin. It goes through the proteasome. Proteasome straightens it. So kind of like when you take those, the pins and straighten it. What is that? Oh my God. The clipper, the little clips that you connect your papers together. And then you separate them to little polypeptides and eventually to amino acids. Your body is very unforgiving of any problems with proteins. So protein misfolds from any sort of mutations. So if you have some sort of mutation in a protein, let me back up. If you have some sort of mutation in the original sequence of the mRNA, it causes misfolding of those proteins and therefore causes a change in its conformation. The reason the 3D structure is affected is because the amino acids, each amino acid has its own characteristic, which means you form different attractions and repulsion on different sections, which eventually forms the 3D structure of that protein. So there are very specific proteins called prion proteins that can form in several conformation. Basically, they can have different forms, if you want to say. And these actually can be passed down as what we call infective agents, which was very new to me when I learned about them as an undergrad. And they basically can be passed down from one animal to another and cause an infection and problem in the new animals. There is several disorders that we have associated with the brain as a result of these prions being introduced to the nervous system, and these misfolding basically expand. One of the ones, and here's a list of it. I don't want you guys to know everything, but I think everybody has heard of at least a couple of these. Alzheimer's disease is a big one, and then Huntington disease, another one, and Parkinson's disease. So again, these are basically associated with misfolding of specific proteins that causes damage to the neurons of the brain, and those damages are irreversible and cause brain damage in either form of Alzheimer, Huntington, or Parkinson's, or medical conditions like prion disease, PKU, dementia, etc. Now, prion disease, there is a specific category associated with the specific misfolding of the protein. So the prion disease I have at the end of this is very specific. They are the ones that first was described in sheep, and then it was also identified in cow. Prion disease can develop or lead to mad cow disease, which was, if you're young, you probably won't remember it, but it was a very big issue, maybe close to 2 decades ago, when there was a chance of those prions causing major infections in humans, and basically humans being diagnosed with mad cow disease. So they were destruction of huge numbers of cattle because they were so worried that it's going to get passed on to the individuals. More than 85 animal species develop similar disease. Again, those diseases can be passed on to us. And it's a result of a specific gene called prion-related protein gene, or PRPN. And again, it is transmittable in its specific conformations. So prion disease, again, was popularly called, but there is a specific version of the prion disease that was called the mad cow disease, which is what I was mentioning. It was first identified in New Guinea, and the reason was for that is because those individuals were basically carnivores. Sorry, not carnivores, cannibals. I was like, carnivals not the right word, cannibals. And these individuals basically would eat a human flesh And that was the way that the medical condition would be passed on from one person to another. So that one that was passed on from generation to generation as a result of consuming human flesh was called the Kuru. Was called a prion disease, but another version of that, which is CJD, is the one that was transmitted from cows to humans, and that was the mad cow disease. How prion cause disease? I don't want you guys to go to a lot of details, but the basic understanding is that we really don't understand what prions do in our body in the first place. But we do understand that if they are mutated, these mutations can cause basically aggregations, and those aggregations can cause damage into the tissue. So prion disease, as I have mentioned already, is a result of improper folding of these proteins, and they can be passed on from generation to generation. So it is unfortunately identified as an infectious disease. Here is how prion can cause damage to the nervous system, which is already something I described. So they can accumulate into the nervous system and destroy your regular cells, which is not a good thing. And the picture that you have here is, by the way, is a picture of a nervous cell that's being destroyed from within by the prion disease. This is the picture we usually utilize for Alzheimer's disease. So that's about it. Please make sure you guys focus and understand the steps on transcription and translation. Make sure what's involved in each of the steps, the protein cofactors, how does the transcription eventually be used, or the product of the transcription eventually be used for the process of translation, and then understand how mutations can affect a gene sequence, or more specifically the mRNA sequence, and therefore cause a major problem in terms of the protein synthesis. So that's about it. If you guys do have any questions, please don't hesitate to contact me. Otherwise, thank you, and I will see you in the next lecture.